You're welcome to Sa Majesty Easy World Science Channel. May God help us as we learn today. Amen. So in our chat today, in our live stream, remember this 3 days live stream is according to my followers' suggestion on my community post. So you follow my community guideline and post to make your own suggestions on the next topic to handle. So today we're going to take a look at cell biology, but I have to copy the link and post on my WhatsApp status so that people can join from there. So in today's class, we'll be discussing cell biology, and it is actually an introduction because it's a bigger aspect of biology. We cannot finish everything, but I bet you that by God's grace, you will learn something great today. Remember to subscribe and share this video and check other science videos here on this channel. There are up to 200 educative videos for you available here. And also book for your own private class. I handle chemistry, biology related courses, then practicals, qualitative and quantitative analysis, industrial production processes. So I make all these services available for you. You can contact me with the number +234 7065166217. If you are calling within Nigeria, you don't need to put the +234, you just 07065166217. Of course, that is it. Now, today, what is a cell? We will start by the definition of a cell. A cell is the basic structural and functional unit of life. In other words, you can say that a cell is actually the foundation of life. The basis upon which life starts is what we call cell. It is the foundation and the building block of life. We call it cell. By extension, to explain what I mean that by the definition that cell is the basic structural and functional unit of life, it means that it is the functions of the cells that gives the organism its functions and characteristics. And this means that organism cannot perform any function that its cell or cells cannot perform. If you don't have any cell that have the ability to perceive light and now transmit it to some parts, you cannot see. That is it. So you are reflecting what your cells can do, and cell is the basic unit of every life. And then to illustrate this for you, it gives us our function. For example, animals, we don't have, our cells lack the ability to convert solar energy to chemical energy, and that is why we cannot produce our own food, because we lack the ability to convert solar energy. Why? Because our cells lacks chloroplast, which has the chlorophyll that have the ability to attract solar energy, and therefore we, the heterotrophs, cannot perform that function. Another example to tell you that the cell is the basic unit and the structural unit and the functional part of life is this. Our eye have the function of perceiving light. Now, what gives the eye is an organ, and inside that organ, remember the organization of life, it must start with cell, from cell to tissue, tissue to organ, organ to system. We are system, we are a complete organism, but we started, everything we are doing depends on what our cells are doing. Now, I'm using the eye, for example. The function of the eye is to perceive sight. It is for the physiology, or the action is just to see. That's the main work of your eyes. Then what gives the eye that function? As an organ, there is a tissue there. In the eye, we have a retina, which is a tissue. Then inside that retina, that's the retina that's the part of the eye that makes the eye perceive light. But there is something that gives that retina its function, and these are cells, and those cells are called the light sensory cells, commonly called rods and cones. So the rods and cones inside the retina of the eye, and the retina is inside the eye. That what determines the function of this eye we have, and that's why it's capable of perceiving. The same thing is applicable to every other organ. Then we can now know that a cell can be a complete living thing. It's just like a wall. What makes up a wall? When you join blocks together, it builds a wall. So what makes up life is a cell. But unfortunately here, one single cell can be a complete living thing, and that's what we call unicellular organisms. But unlike joining blocks, one block cannot make a wall, and neither can it make a house. But here, a single cell can be a complete organism. Then, cell being the basic structurer, like some organisms have a shape similar to the shape of their cell. For example, Paramecium caudatum have the slipper shape, and what gives the organism as a whole its shape? The shape of the cell. So amoeba don't have one particular shape because of their ability to change their shape, form the pseudopodia. Therefore, they are defined as not being definite. That's, they have indefinite shape. Why? Because the cell don't have the ability to take one particular shape. So the structural and the functional unit of life is cell. Now, let us now take a look at brief history of cell. The cell was discovered by the man. Okay, I think it's time I write. Remember to ask your questions because I'm here for you as you are there for me. Thank you for joining. For those of you live, I really appreciate. You can share the links with your friends and your partners. And remember, the live stream will still be available even after. Okay, then what we have, our topic today is cell biology. That is actually cell. I've defined cell. What is cell? You say a cell is the basic structural and functional unit of life. Then we are discussing, let us take a look at a brief history of cell. How and who discovered cell, when and how. The word cell was coined and also discovered first by a physicist. Imagine that partnership in science. We are discussing biology, but a physicist discovered cell. Some authority might erroneously tell you that the man is a biologist. No. Cell was first discovered by Robert Hooke. If you hear Hooke's law, Hooke's law in physics, he discovered cell in the year 1665. So I want to give you the brief history on how he discovered the cell. In Robert Hooke, being a physicist, is embarked. He made a compact microscope by himself, the light microscope. So on the attempt, maybe I will call it curiosity, trying to check the efficiency of the microscope he constructed. He observed a thin slice dead cock tissue. That's a slice of a dead cock tissue under the self-made microscope. He discovered that this tissue from the dead cock, that's the cock tissue, is made up of neatly arranged chambers that is enclosed by cell walls similar to the cells of a honeycomb, because the word cell has been existing in English to represent the tiny holes found on a honeycomb. So he decided to call each of these chambers he discovered in this slice of dead cock tissue a cell. He did not discover any living content of the cell. Besides, he used a dead cock tissue. That's why I tell you, this is why he was unable to discover any living content of the cell. But rather, in his observation, he discovered something like this. Just trying to that there are chambers, and each chamber is enclosed by wall like that of a honeycomb. Honeycomb is where the honey deposits their where the bee deposits their honey. So the honeycomb, each of the tiny perforations in a honeycomb is also called cell, and he now attributed it to this. Now, being a physicist, he did not further ahead. He did not go further based on this because he's not a biologist. Then with time, other cytologists Please, the scientific study of cell is called cytology, and specialists in the study of cell are referred to as cytologists. They are actually biologists, but the special area should be in the aspect of cell, because the prefix CYT is for cell. Anything CYT is talking about cell in science. Cytology, cytoplasm, cytokinesis, anything cyte, leukocyte, that is one thing to understand about this cell. We are using the prefix CYT for cell. Then the scientific study is cytology. Then the specialties, the expertise, those people that are specialized on cell are called cytologists. Then other cytologists now helped. In 1831, another remarkable discovery was made. In 1831, the same robot, maybe if you answer robot, you will become a scientist. You can take it. I think most of them are robotic in name. Robert Brown discovered the nucleus of cell. 1831, Robert Brown discovered the nucleus of a cell. He made that discovery as a botanist. Robert Brown was a botanist, and he made this discovery, finding out that in all the plants there is the tiny central dot he referred to as the nucleus. Remember, Hooke did not discover anything. Then Robert Brown was a biologist, and other of his impacts in biology, apart from this, is that he was the first actually to differentiate between flowering and non-flowering plants, of course, the angiosperms and the gymnosperms. Then it might also interest you to know that he is more popular in chemistry than in biology. Like the name, actually, if you hear about Brownian motion used in chemistry, physics for the kinetic theory of matter as evidence to support it, it was actually this man that propounded that Brownian motion, where he accidentally discovered the haphazard movement of the pollen grain of and the spores of some plants suspended in water. He now propounded that Brownian motion as one of the evidence to support that every matter possesses kinetic energy, but that's a separate thing altogether. Then in 1835, of course, in 1835, in the year number 3 remarkable thing, 1835, Felix Dujardin, I guess, yes, studied and discovered that there are living contact, that the cell has living units, and he called the living part of the cell the sarcode, or the sarcode. The sarcode was to represent the living part of the cell. This sarcode was later named protoplasm. It was later named protoplasm by von Mohl, probably in 1848, of course, von Mohl. So von Mohl called the sarcode protoplasm, and they all have the same meaning the living content of a cell. Collectively with even Purkinje, they called this sarcode protoplasm. Then in 1838, a German botanist, yes, of course, by name Matthias Clairaut. So Matthias Clairaut, I might not be pronouncing their name perfectly because these are not my own language. So the Matthias Clairaut, now a botanist, also confirmed that most plants are composed of cells. He was able to prove that many species of plants have that constant chamber that Hooke observed from the cork tissue. Then the subsequent year, being 1839, another German physiologist by name Theodor Schwann, of course, Theodor Schwann now also reaffirmed and confirmed that animals are also composed of this cell that they are talking about in plants, that they are also found in animals. So these discoveries, then subsequent things were discovered. Von Sachs were able to discover the chloroplast, that's the chlorophyll containing pigment, which is the chloroplast, that's the von Sachs. And then chromosomes were discovered by Walter Fleming around 1882. So many of them made now impact. Rudolf Virchow was able to predict cell division, of course, but cell division was actually discovered and described. Mitosis was first described maybe in a reasonable level by Fleming. That's Walter Fleming in 1882. So it was in the course of the description of the cell division that he discovered chromosomes. Although it wasn't him that named it chromosome, it was Henry that actually called it chromosomes. So that is the little history of cell that is necessary here. These names I'm calling will help you know who and who are actually cytologists and their contributions, of course. Now, these big discoveries and the contributions of the great scientists, in the person of Robert Hooke, the discovery in the person of Dujardin, the Pockinger, von Moe, Matthias Clairaut, Theodor Schwann, you call them, named Rudolf. All these things were now, it gave basis to a theory known as cell theory. All these, their discoveries were a bit summarized by a theory called cell theory. So what did the cell theory say? Some might give you the cell theory in 4 sub-statements, but the cell theory I'm going to give you here, the 3 basic ones. So the cell theories were a kind of the summary from the discoveries of these great cytologists. So these things were summarized as cell theory. Cell theory should be discussed under 3 headings. The first statement under cell theory is that every living thing is composed of cell or cells. That's the number 1 statement one should know about the cell theory. Every living thing is composed of cell or cells. Then number 2 statement there is that's the definition of cell now. Cell is the basic structural and functional unit of life. Then the number 3 statement under the cell theory is that cells arise from previously existing cells. In other words, you say that cells beget cells, probably through cell division, as confirmed and explained properly by Walter Fleming. So in the cell theory, one should just say that all living things are composed of cell or cells. Then secondly, cell is the basic unit and basic unit of life. Then thirdly, cells arise from previously existing cell by cell division. So these are the basic statements that summarizes cell theory. Then we have to take a look at the cell within us. Like you, can you isolate any cell in you? Not just by far abstract learning. In you, in the introduction, in the beginning, I was able to mention a cell in us and the function. I called rods and cones. Can you mention any other cell in you? Okay. Try. Okay, did I hear you say red blood cell? Yes, of course, it's a cell. The white blood cell, they are all cells. The neurons are cells, of course. The muscle cells, there are numerous of them. We have not less than billions of cells in us, although it started from 1 cell. That should be a new topic altogether, cell division, of course. But we are here just introducing the cell biology or cytology as it may concern you. Then, do you have any example of plant cell? You say the word plant cell. Can you give anything that is a plant cell? Okay. You hear about the gut cells, the epidermal cells? These are all plant cells, of course. So both plants and animals have cells, and that is the basis. So we have brought it closer to ourselves so that we can be able to know what we are doing today. Now, let's take a look at types of cells. We want to classify cells. The next thing are types of cells. Types of cells. Not here, that's where there is a big contradiction. Some might just tell you we have 2 types, which are plants and animal cells, okay? But I wouldn't be buying the idea that way. Cells can be largely classified into 2, which will be prokaryotic cells and eukaryotic cells. It is now under eukaryotic cell that you can now find plants and animal cells. Because if you say plants and animal cells only, where will you place bacterial cell? Definitely, bacteria is not seen as a plant in the 5 kingdom system of classification. That's the bacteria belong to the kingdom Monera. Therefore, if you say we have 2 types of cells and just say plants and animal cell, I'm now asking you, where will you place bacterial cell? So that's why normally there are 2 main types of cell, and that should be prokaryotic cells and eukaryotic cells. And this classification also gives us the two empires of life. That's the basis from which life is divided into two empires. You know, we have levels of classification. In taxonomy, we have kingdom, phylum, class, order, genus, family, genus, species. But there is a rank, a taxon above kingdom, and that should be domain. Then after domain, you have empire, which means we have empire, domain, kingdom, phylum. Therefore, we have two empires of life, which are actually the prokaryote and the eukaryote. And this is the basis by the cells, by the two types of cell we have. I said it should be divided into two, the prokaryotic cells and the eukaryotic cells. Karyotic cells and the eukaryotic cells. Now, the etymology, or to describe this term, anything pro is a kind of before, then karyo, as far as this topic is concerned, karyo means nucleus. EU in science means true. Then karyo, it is still nucleus. So if you can even understand the etymology or the foundations of the words, you can describe what is prokaryotic and eukaryotic. Pro, before, proceeding, then karyo means nucleus, then U means true. Then karyo still means nucleus. So the cells can be divided into prokaryotes and eukaryotes. And under these prokaryotes, that's where we can find the archaea, bacteria, and then the eubacteria. So over here, that's just the bacteria, makes up the prokaryotic. Then on this eukaryotic here, you can now find your plant cells, find your animal cells, fungi cells, agar cells, animal cells, fungi cells, because they are not exactly the same. So plant cells, animal cell, fungi cell, and the algae cells are all still under the eukaryotic cell. Before we go down here, let's handle the prokaryotic cells and the eukaryotic cells. Now, what are prokaryotic cells? A prokaryotic cell is a cell that lacks nuclear envelope. In other words, that lacks true nucleus. Maybe those organisms existed before the evolution of true nucleus, and therefore their name is a kind of eukaryotic, meaning their name is a kind of prokaryotic before the evolution of true nucleus. That is from where they gain their name. Okay, then by definition, I've told you they lack nuclear membrane. Not that they lack nuclear content. They have nuclear content, but they lack the membrane. The content of their nucleus is mixed with the content of the cytoplasm. While eukaryotes are actually cells that have true nucleus, that have well-defined nuclear membrane. So it is the nucleus that is the yardstick for this group in prokaryotic and eukaryotic. In other words, some people will tell you this one have false nucleus, while this one have true nucleus. What is a false nucleus? A nucleus that have a nuclear membrane. What is a false nucleus? A nucleus that lacks nuclear membrane, that is without an envelope bounding the content. While a true nucleus is a nucleus that have a clear nuclear membrane. So if I draw a cell here and you see something like this, you just see nuclear content without seeing the membrane. This is a prokaryotic cell. But if we have a cell in this way and you have a membrane covering the nucleus, covering the nuclear content, this is, if you have nuclear membrane, this is automatically a eukaryotic cell. But this one that lacks the nuclear membrane is called prokaryotic cells. So this is the major thing that distinguishes prokaryotes from eukaryotes. But there are other differences, of course. There are other differences between these these two types of cells. It's not only based on their nucleus, on the nature of their nucleus. You can see the differences, but before I give it, it is good I take a look at the structure of the so-called prokaryotic cell. That's, I might illustrate a typical bacterial cell, which is representing other prokaryotic cell. A typical bacterial cell is what I want to draw now to represent prokaryotic cell. Before we now go to the eukaryotic, a typical bacterial cell. So a bacterial cell will have the invaginations, and then the cell membrane, of course, then the cell wall, and then the capsule, which may be present or absent. Then there might be flagella, present or absent. Then there is a nuclear content, of course. Then you have other aspects. Here is just to represent the nuclear content. You have the ribosomes scattered all over. So they are very, very simple cell. Okay, by labeling, here is our cell membrane. Here is the cell wall, which is made up of peptidoglycan layer. Then here is the capsule or the slim layer. The capsule, it might be present or absent. Slim layer, it might be present or absent. These two is not compulsory. Then these invaginations inside is what we call the mesosome. Then here, the nuclear material without, these are the nuclear material. There is no nuclear envelope. So the nuclear material without nuclear envelope, the region, this region can be referred to as nucleoid. Just those things having the chromosomes without it. Then this is the flagella, which might be present or absent. Flagellum is singular, flagella is plural. So this is a typical bacterial cell, of course. Now, looking at this, we can now distinguish it from eukaryotes. For example, I guess if you can screenshot, because I will clean this. I'm running on a small board. I'll still draw plants and animal cell when I face eukaryotes squarely. Then, of course, you should know that this is the cytoplasm. Then the dots inside is just the ribosomes. The ribosomes are the dots there. Okay. If you have questions, you bring it alongside. I am here for you. And remember, this is Her Majesty Easy Word Science channel. If you are just joining us, we are discussing cell. Of course, that is the scientific study of cell. The definitions I've given you that one. And also remember that this is the introduction. We cannot live without cell. And cell, like I said, is the basic structural and functional unit of life. I've given you the cell theory. I've called the cytologists and their contributions. And this cell we're talking about was actually discovered by a physicist by name Robert Hooke. Then he couldn't describe anything there because he's not focused on the study of life. He's a physicist, of course. Then remember to book for your own private classes if you want to know more about biology, chemistry, and the biology-related courses, chemistry-related courses, your anatomy, biology in the beginning aspect of it. You contact me, of course, in the comments box, or you get me on WhatsApp +234 7065166217 +234 7065166217. So that should be on WhatsApp, or you can visit my site easyworldscience.com. Yeah, www.easyworldscience.com. Remember, I also make laboratory equipments available, laboratory apparatus. I also help you build your laboratory starting from the scratch. You can also hire me to teach your students chemistry and biology online. Distance is not the barrier. There are many people I'm handling and they are doing well. So don't be left out. Now, I want to give us a comprehensive differences. The comprehensive differences between prokaryotes and the eukaryotes. I'm going to clean this typical. Remember this mesosome. This invagination is called mesosome, which is absent in eukaryotes. The so-called capsule slim layer, they are not present in eukaryotes. Okay. So we want to see the differences between prokaryotic cells and eukaryotic cells. So number one is that nuclear membrane or envelope is absent. That gave them the name. Nuclear membrane is clear and well defined. Nuclear membrane is present. Then another difference is that mitochondrion absent. Here, mitochondrion as the powerhouse of a cell, present. Then here, mesosome, present. Here, mesosome, absent. The mesosome is absent in eukaryotes. Then chloroplasts and other plastids, absent in prokaryotes. Chloroplasts and other plastids, absent. But here, chloroplast, present. Chloroplast and other plastids are present, except in animal cells and other plastids are present, except in animal cells. So animal cells don't have plastids. Then there are other differences. Number 5, we'll take a look at the contents of their cell wall. You now listen to me. The contents of their cell wall. The cell wall of prokaryotes, mean the cell wall of bacteria, is actually composed of peptidoglycan. Yes, the peptidoglycan layer, that's the contents of the cell wall of prokaryotes. Then the cell wall of eukaryotes is either made up of cellulose or cellulose or chitin. Yes, fungi cells have chitin, or what you might call chitin. That's chitin, chitin, or cellulose. Plant cells have cellulose, while fungi cells have chitin, but they lack peptidoglycan. These ones, the bacterial cells have the cell wall. I'm describing the cell small, but it is filled up, so I have to be describing for you to understand. So what I pronounce is peptidoglycan. So the cell wall of prokaryotes have peptidoglycan layer, while it is absent here. The cell wall of eukaryotes is either made up of a chitin or cellulose. Like that of plant cells has cellulose cell wall, the fungi cells has nothing but a chitinous cell wall or chitinous cell wall. Then, if you come to the nature of the distribution of ribosomes, you can also distinguish them. The distribution of ribosomes can distinguish prokaryotic cell from eukaryotic cells. The ribosomes are distributed in the cytoplasm of prokaryotic cells in 70S. There is a unit used to measure the sedimentation rate of the ribosomes, and this is Svedberg, or I don't know how you pronounce it either. So this is the unit SVEDBER for measuring the sedimentation rate of ribosomes. That of prokaryotes is in 70S, while that of eukaryotes are normally distributed in the endoplasmic reticulum in 80S. This one is 70S. This S means Svedberg. So the distribution in this is 70S, while here is 80S. And if you come to this distribution, you have large subunit and small subunits. In ribosomes, the 70S is made up of actually the 50 large subunit and 30S small subunit. 50S large subunit and 30S. Though I know if you sum it, it will give you 80. That's not the way. It is 70S, but here is 80S. In terms of the distribution of the ribosomes in the sedimentation rate, using this Fedberg as the unit, that of eukaryotes is actually distributed in 80S, which is in 60 large subunits and 40 smaller subunits. So that's, you can use their distribution of a ribosome to differentiate them. Then you can also differentiate the prokaryotes from the eukaryotes. Which other unit? There may be other criteria that can distinguish them. Okay. You can also take a look at the recognition sites in the promoter area of the DNA of a bacteria. Is that of bacteria is actually in what we call the Pribnum box. This one is in the recognition site in the DNA. That is where protein synthesis, the transcription starts under this for bacteria. Bacteria is using Pribnum box. The Pribnum box is composed of what we call TAT. TAT is 6 of them, meaning TAT. That's where protein transcription starts in the bacteria, is the Pribnum box. That is Pribnowy. This one is what we call Hogness box. That is for the eukaryotes. Although there is a little thing here, the Hogness box is what we call Tata in Igbo. Funny enough, it looks like Tata in Igbo means today, but this is thiamine, adenine. That is the point where the protein transcription takes place, while this one is in the prime box, which is called at the sequence of -10. Sequence -10. This T is still thiamine, adenine, thiamine. Then, but the archaea or the archaea bacteria also belong to the prokaryotes, but they also use the Hogness box, just like the eukaryotes. It is only the bacteria that uses this Pribnum box. So it is not a clear distinction. This box for the recognition site in the promoter site of their DNA is not clear, because archaea bacteria uses TAT, just like eukaryotes, while the so-called bacteria only, that's the eubacteria, uses the TAT, known as the Pribnum box, for the transcription to start. Is it clear? Okay, that's great. Thanks for being there. For some of you that are still online, that's wonderful. You are doing great. Okay, then these are the little differences for now. I can remember. There are other, even the endoplasmic reticulum is absent. Yes, endoplasmic reticulum is absent in all prokaryotes. It's present in the so-called eukaryotes. So having handled the prokaryotes, it's time for me to look at the eukaryotes very well. The eukaryotes are those of them that have the true nucleus, which is a nucleus with nuclear membrane that envelopes it. And you can see the examples under eukaryotic cell. That is where you can find plants and animal cells. Now we have to take a look at a typical plant cell and a typical animal cell. We will compare and contrast. Then after that, we go into the cell structure, general cell structure description. Understood? So when they tell you types of cell, I wouldn't know what they want you to answer. If they say 2 types of cell, normally it should be prokaryotic and eukaryotic cell, but most often your examiner wants you to say plants and animal cell, and I've proven you wrong. That when you say plants and animal cell, where will bacteria cell belong? Where will fungi cell belong? Remember, in 5 kingdom system of classification, that's the most one we use commonly. You have monoreal kingdom, the protista, the fungi, animal, then plant kingdom. So when you say plant and animal cells, does it mean that the monorans, the protists, and the so-called the fungi don't have a cell? So if they tell you types of cells, you say prokaryotic cells and eukaryotic cells. Then the eukaryotic cell, that's where you have the plant cells and animal cells found there. So I'm going to consider the plant cells and the animal cells. Plant cell, a typical plant cell and a typical animal cell. Typical means that there is nothing exactly like when we use the word typical, it is trying to tell you that you don't expect one particular plant cell to look exactly. It's just a generalized plant cell. That's the meaning. A generalized animal cell. Don't you know that, for example, if you come to the drawing of any cell, have you seen any cell with tails? But there are cells with tail. For example, the sperm cell in man have tail, but it's not part of it because tail is not one of the generalized structure found in most cells. So there are some features omitted, and hence we use the word typical, meaning generalized. It's just like something that is expected to be present in average cell. So it's not like I'm drawing an onion cell, mango cell is a kind of the typical representation. So in the drawing of the plant cell, if you are finding it difficult, I'll give you the cheat. What you're going to do is just make a sharp rectangle, can cut off the edges, curve it, curve it. So then you have to place your cell wall. This is the cell membrane. Hope my camera is getting my drawings. Okay. Then we have the cell membrane of adjacent cell because it's a kind of isolated one. So this thing I'm drawing is just trying to represent another cell, like if there is another cell. Because some of you might not know why the three lines. The three lines doesn't mean there are other layers. No, this is just the cell membrane of another closed cell because it is assumed that this is not an isolated cell completely. But if you remove these lines, you have no crime. Okay, then you draw your vacuole. For plant cell, you should make it the largest organelle. Then you should now draw your nucleus, try to surrender it with your endoplasmic reticulum. So then the ones attached to the nucleus actually have the ribosomes, and that makes it rough. You should make it rough. Then insert your nucleolus, chromatin thread. Then draw your mitochondrion. We represent it with spiral line. The number you draw is not a problem. In exam, it's just to draw something you will show that it is present. Then you can draw your chloroplast. This is the way to represent it. Then you also have your free endoplasmic reticulum lying. You have the Golgi apparatus. So the number of this you drew, there is no limit. Just draw the number that will show the features present. Then you saturate here with the ribosomes scattered in the cytoplasm, of course. You can make these two so that we can differentiate rough endoplasmic reticulum from smooth. The word endoplasmic reticulum means like net, something derived from net. Okay, this is a typical plant cell. Then we forge ahead. In that of animal cell, it's not that rigid, so you can get something like this. Then draw your nucleus, the nucleolus, chromatin thread. Then as usual, And we have the centrioles, which is present, and put the lysosomes. Same thing, Golgi apparatus, Mitochondrion, the endoplasmic reticulum, okay. This is a typical representation of the plant cells and the animal cells, of course. Okay, then we will have to consider some things, the differences between the two and the similarities between the two, or labeling either. Because of time, I will number first. We have to identify this. This is 1, then this one is 2, then the center here is 3, and this membrane is 4. Here is 5, and here is 6, and here is 7, then here is 8, here is actually 9. Okay, these are the basic things we need to know. There are many other parts. Then number 1 is the chloroplast. Number 2 is the nuclear membrane, which makes it a prokaryote, of course. The number 3 is the nucleolus. Then number 4 is the nuclear membrane. That's the nuclear envelope. Then number 5 is the mitochondrion, which is the powerhouse. Then number 6 is the vacuole itself. Then number 7 is the membrane enveloping the vacuole, and the membrane covering a vacuole is called tonoplast. So number 7 is the tonoplast. Tonoplast, tonoplast. Then number 8 is the Golgi apparatus. Then number 9 is the cell membrane. The cell membrane, which is the same thing here. If you don't call it cell membrane, you can call it the plasma membrane or the plasma lemma. The plasma membrane or the plasma lemma, all of these two terms stands for one thing, which means cell membrane. Okay, then the labeling for the basic parts is being settled, but know that 2, 3, and 4 actually represents the nucleus, of course, because 2 is the nuclear membrane, 3 is the nucleolus. Then I'm omitting another part. This is the chromatin thread, of course, those things inside. Okay, so having seen these typical plants and typical animal cell, you can make a difference. What are their differences? Let's take a look at differences between plant cells and animal cells. You can easily read it up. I know these are easy things you can do. So that makes me take you to the next level, cell structure, because differences between plants and animals, they are very easy and cheap. So what I just did now is just to teach you the cheats on how to make this drawing perfect if you are faced with it in the examination. Okay, so I guess I'm ready to go. Remember, there is no large vacuole here. There is no large vacuole in this cell, but there is large vacuole here, and that is for plant cell. There is no cell wall here, but there is cell wall here. At least that's enough to make a difference. But both of them have mitochondria, Golgi body, endoplasmic reticulum, nucleus, nucleolus. So these are their similarities, although indirectly I've done that I said I'm not going to do. But thank you for being there so much. So let us see cell structure or cell components. The components of a cell. I want to draft a way that will make it easier for us. Components of a cell. One can divide it into 3, which are actually, you have the cell, let's make it snappy here. Okay. Okay, we want to see that there is the protoplasm, yes, of course. According to von Moe, that's the circle discovered by the so-called Purkinje. Then we have the cell extensions. Cell extensions. That is the so-called example here is the flagella. Here we have also the cilia. Then we also have other coverings. That's how I did it. other surface coverings, that is where cell wall comes in, because cell wall is not part of the protoplasm. Here we have the cell wall, we have the capsule, the slim layer, most of them. Then if you come to the protoplasm, you divide it into two, which is actually the cytoplasm and the nucleus. Simple. Then inside the nucleus, we also have there is a part that is missing. Okay, yeah. Inside the so-called cytoplasm, we can divide it into organelles and cell inclusions. Although some doubt that cell inclusions shouldn't be part of protoplasm because it's not the living part of a cell. Inclusions, cell inclusions, and then the cell organelles. Then the cell organelles. Then when we talk about the nucleus, we talk about the nuclear membrane, the chromatin, the nucleolus. Yes, these are the components that makes up the nucleus. Now, in this aspect, you might have come across or hear some of these things, except one. The cell inclusion is where I want to lay emphasis on. The cell inclusions are either secretory granules or granules that were absorbed, that is engulfed granules, or secreted granules inside the cytoplasm that is not a constant part of the cytoplasm, of course. So they involve the cell inclusions includes the so-called secretory granules such as starch granules, the glycogen granules, then the pigments, like when we talk about pigments in plants and animals, they are part of cell inclusions, of course. They are not actually part of the cytoplasm. They are either produced to be exported or something that was actually imported into the cell, either by endocytosis, yes, of course. So the cell inclusions has been settled. Now, when we come to the cell organelles, these are the minute structures that perform a particular function for the cell. So under the cell organelles, that is where I will lay emphasis on. So the cell organelles, we first of all have the so-called the cell membrane. Yes, the cell membrane is a selective membrane that covers the cytoplasm of a cell. It is semi-permeable and it is flexible. It's not rigid, unlike the cell wall. Then the cell membrane is a double lipid bilayer. That is a lipid bilayer that is made up of the lipoproteins that has a head and a tail. So when you take a look at the heads and the tails of the so-called cell membrane, what I'm just trying to tell you here is that the cell membrane you see we draw with one line has double layer. It's a double layered membrane, and most all the biological membranes have double layers. So I want to take little time now to explain the double membrane. That's the lipid bilayer, the lipid bilayer of cell membrane and other biological membranes. When I say other biological membranes, I mean the membranes covering other organelles such as the chloroplast, the mitochondria, even the nuclear membrane has this anatomy I want to describe now. So let's take a look at the anatomy of the cell membrane. So the cell membrane has a double layer. In that double layer, we have a hydrophilic head and then a hydrophobic tail. Another layer will do that way. So here is the head, here is the tail. The head points outward and inward such that the surface is the outer and the inner surfaces of the membrane is made up of the hydrophilic head. That is the head that loves water, and they create a barrier, a water repelling center, because these tails don't like water. They are water hating, of course. Okay, so in the labeling here is the hydrophilic head. I'm taking a look at the anatomy of the cell membrane, the lipid bilayer. the lipid bilayer, that is a layer that is made up of two lipid layers. Of course, bi means two. So this is hydrophilic head, which is polar. This is polar in nature, of course. Then these ones are the hydrophobic tail, water hating, hydrophobic tail. So this is the way when you see cell membrane, this particular one line I drew is in this case, such that this one is pointing inside, this is pointing outside. So on the surfaces is made up of the hydrophobic heads, and then the inside, the barrier in between the two heads are the tails, which forms a water repelling layer, of course. Then with time, we also have the transmembrane proteins, which helps in active transport, such that this thing continues in this way. So we might not have time to continue all this, but this is just to tell you how the cell membrane looks like. It's a lipid bilayer. It's made up of, is a double layer, just like other biological membranes. And this is the description, the anatomy of it. The tails are hydrophobic, the tail, meaning water hating, whereas the heads are actually hydrophobic. And that means that any molecule that is not soluble to lipids will not be allowed to pass. For them to pass, they need a secondary, or what we call second messenger. At a stage, you understand what I mean by second messenger, of course. But just know that the cell membrane is made up of a lipid bilayer, and this is the anatomy. Then transmembrane proteins are there, which helps in the transportation of some material, bulky materials, of course, in the so-called exocytosis, endocytosis. Most times, or active transport, they use the transmembrane proteins, and also in facilitated diffusion, these transmembrane proteins or surface membrane proteins play a very big role. Now, what are the functions of the cell membrane? The cell membrane, number one, allows the selective movement of materials in and out of the cell. Remember, another name for cell membrane is plasmalemma. Plasmalemma in case of exams. So PLASMA, LEMMA, plasmalemma. I said, one, it allows the selective movement of materials in and out of the cell. Two, it delimits the contents of the cytoplasm. In other words, it controls the contents of the cytoplasm. It ensures that what's supposed not to go out will not go out. What's supposed not to come in will not come in. That is it. And that is why the cell membrane plays a role in osmosis, diffusion, allowing molecules, permitting molecules of only selected sizes to pass. So that makes the cell membrane, gives it the ability to limit or delimit the content of the cytoplasm. Then there are surface proteins on the cell membrane that may act as immunological identification or biomarkers. In the case of histocompatibility, they are just the cell membrane sometimes bears what identifies a cell. That is it for you. Then after the cell membrane, we'll take a look at the so-called the cytoplasm, which is actually the largest part. So the cytoplasm is nothing but the space between the nuclear membrane and the cell membrane. So the space and the fluid that is enclosed by the cell membrane is called cytoplasm. And then inside the cytoplasm, there is what we call the organelles are suspended inside it, and there is a fluid inside the cytoplasm known as cytosol. So cytosol, otherwise called the cytoplasmic fluid, is the fluid inside which the cell organelles, meaning the Golgi apparatus, the endoplasmic reticulum, the mitochondria, are all suspended in the cytoplasmic fluid, otherwise known as the cytosol. Then the cytoplasm also have fibers that is called the cytoskeleton. So the cytoskeleton is a kind of fiber that is made up of microtubule, microfilaments, and intermediate filaments. So the cytoskeleton, that is the fiber that gives rigidity to the cytoplasm. It has many functions. It actually supports the cell membrane. Then it supports and also provides anchorage for the cell organelles. Then when it comes to what we call the ectoplasm and endoplasm, in the ectoplasm, the denser part of the cytoplasm, like when we talk about the paramecium, caudatum, or the amoeba, there is what we call, their cytoplasm is divided into denser and less dense. The denser part is the outer part, which is filled with much of this cytoskeleton. Is a kind of the framework of a cell, the framework of a cell, made up of microtubules, microfilaments, and intermediate filaments. That is cytoskeleton. Then, in the ectoplasm, it helps in movement. So cytoskeleton helps in movement, especially it supports the cell extension, that is the flagella, the cilia, in movement. Gotten that? Then, in the aspect of that the so-called cytoplasm, the cytoplasm has a fluid known as cytosol. The function of this cytosol is that as it is moving, the movement of the cytosol, that is the cytoplasmic fluid, together with the content of the cytoplasm is what we call cytoplasmic streaming. Cytoplasmic streaming, and this streaming brings about exchange and interchange or transport of material within the cell. So this is now transport within the cell is affected by cytoplasmic streaming. So when we talk about transport in unicellular organism, one of the things that makes it effective and possible is cytoplasmic streaming. Then the cytoplasm is sensitive. It responds to the red acid dye. It picks the acidic ions in red acid dye known as eosine. So in this fixing and staining, that is in cytology, of course, you must involve the use of microscope to study cells because most of these cells are actually microscopic. They are not visible enough with the eye, and then the cytoplasm is a bit transparent, and that makes it impossible for you to visualize the content very clear. So in the aspect of microbiology, before you observe minute structures, microorganisms or tiny organisms or small cells, you must stain it. So staining is just addition of dyes to color it to enhance their visibility under microscope. So when scientists want to study the cytoplasm, they actually try to fix it with a red acid dye known as eosine. This eosine is a red acid dye and is acidic. This eosine is only sensitive to the cytoplasm. It's not sensitive to the nucleus. So you can differentiate the nucleus from the cytoplasm using this eosine. The cytoplasm is sensitive to the red acid dye, and therefore we can say that the cytoplasm is the acidophilic part of a cell, unlike the nucleus, which is the basic part, which will respond to the basic dye, the methylene blue, of course. So that's just by the way. Then you can also, in the case of analysis of samples from, you can preserve the cytoplasm of a cell using the Fleming fluid fixative, or actually you use what we call post chromatization using potassium heptaoxodichromate 6 together with sodium chloride solution. You can preserve that. That is when we go to actually histology. Remember, histology is the study of tissues, and it is not easy for you to just single out cell and study it without the tissues. Okay, then we move to the let me just describe the functions of the so-called cytoplasm again. It suspends the other cell organelles. Cytoplasm suspends other cell organelles. 2, cytoplasm contains a fluid, cytosol, and through the streaming of this fluid, that brings about exchange of materials within the cell. Exchange of material within the cell is affected by cytoplasmic streaming. Then, which other function? The cytoskeleton provides anchorage and support for the cell organelles and also the cell membrane. The cytoskeleton of the cytoplasm helps in movement, especially in the ectoplasm. Yes, so these are the common functions of the cytoplasm. Then the part again that we need to consider is the nucleus itself. The nucleus of a cell is the control center of a cell. The nucleus, remember, that's the most vivid part of a cell if you should observe it under microscope, of course. Then I said it is the control center, and it was discovered by Robert Brown in the year 1831. And then the nucleus is composed mainly of chromatin thread, which will later form the chromosomes during cell division, as it was described first by Walter Fleming in 1882, of course. Then this nucleus have also nucleolus. So chemically, the nucleus is composed of the chromatin thread, which is actually DNA. So the nucleus is composed of the DNA, deoxyribonucleic acid. That's why the word nucleic, nucleus. nucleic acid come into play, meaning it is produced in the nucleus. Then the nucleolus is the center of the nucleus. The nucleolus is made up of RNA. I repeat here. So if this is a cell and this is the nucleus, then over here we have the center. This center is the nucleolus, which contains RNA. While here, outside the nucleolus is mainly the DNA, which is the chromosomes. Then the membrane bounding it is what we call actually the cell, the nuclear membrane, of course. So by function, I've just described the nucleus. But by function, I already said it is the control center of a cell. It controls every other activity of the cell. How does the nucleus control other activities of the cell? By controlling protein synthesis. How can it control protein synthesis? Because it has DNA. So it is this nucleus that controls other activities of the cell. Then again, it is this nucleus that initiates cell division. So number 2 function is that cell division starts from the nucleus. Cell division starts from the nucleus, which means the origin of reproduction by cell division starts from the nucleus. Then thirdly, it is the nucleus that contains the genetic material. It is the nucleus that is responsible for the transfer of character from parents to offspring, of course. So it contains the genetic material DNA, which is still responsible for transferring character from parents to offspring. The nucleus also produces RNA, of course, in the nucleolus. So these are the basic functions of the cell nucleus. Remember, if they tell you the control center of a cell, you say it is the nucleus. Then we take a look at another organelle that is very important we consider. We can see the mitochondrion. The mitochondrion is the powerhouse of a cell. It is a sausage-shaped structure which made of the cristae, the inside part and the outside part. The inner part is the cristae, but the major function of the mitochondria is to support cellular respiration. It is in the mitochondria that we have most of the metabolic pathways taking place, like the Krebs cycle, that's the TCA cycle, all occur in the mitochondria, in the membrane of the mitochondria, and we call it the powerhouse. There are also traces of DNA in the mitochondria. We call it the mitochondrial DNA. Of course, it has its own function also, but the actual thing for the mitochondria is that it is the powerhouse of a cell. That is the site of cellular respiration, where ATP, that is adenosine triphosphate, is produced or synthesized. Okay, then I said it's a sausage-shaped structure. Another thing we'll take a look at is the chloroplast and other plastids. Yes, chloroplast and other plastids. We'll talk about plastids, Plastid, in plants, not in animals. We have 3 types of plastids. They are 1, chloroplast, 2, chromoplast, then thirdly, leucoplast. Okay, so we are describing plastids. Inside the plastids, we will see the 3 types of plastids are chloroplast, which is the common one and was discovered by von Sachs, and it contains the chlorophyll. Then we have the chromoplasts, and then we have also the leucoplasts. Anything leuco in science or in biology specifically means colorless. So the plastids are into 3. The chloroplast is a plastid that contains chlorophyll, and chlorophyll is the greenish pigment that absorbs light. It absorbs solar energy. There are different regions it absorbs, of course, but the region is not what we're going to discuss here. Just know that the chloroplast is a plastid that contains the greenish pigment chlorophyll, and remember the word chlorophyll comes from the Greek language. Chloro means greenish or greenish yellow, then phyll means leaf. So literally, chlorophyll just means nothing but green leaves. Imagine, but here it means a pigment that absorbs solar energy. We have different types of chlorophyll, chlorophyll A and B. Then this chloroplast is found in the parts of the plant that is greenish in color and is usually exposed to sunlight. What's the function of the chlorophyll? I've said it's to absorb light. Therefore, chloroplast is the site of photosynthesis. Is the site of photosynthesis. That is the part of the cell where solar energy can be used to produce food by combination of CO2 and soil water, and this process is what we call photosynthesis. Then chromoplast, which means where can we find chloroplast? Sorry, we can find it in the green leaves, the stem, the exposed part. Any greenish part of a plant is going to be containing the plastid, chloroplast. Then chromoplast are other plastids that is colored but not green. Like we have the yellow, the red, they are under this chromoplast. So if you have a leaf having a different color apart from green, the plastid responsible for that color is chromoplast. Then leucoplast is the colorless plastid. That is the plastid that don't have any specific color, that don't have color, of course, because white is not a color in science. So colorless plastids, and they are found in roots. They are found in roots. Then from my own observation, I discovered that these plastids can change. I discovered that when you cover a greenish part of a plant for a very long time, that greenish part, the green color will fade and become whitish sometimes, which means I am giving a hypothesis that maybe excluding light from the parts that have chloroplast will lead to the conversion of chloroplast to leucoplast. It's possible. And sometimes I discover that if you expose some roots, which suppose not to be greenish, normally roots have leucoplasts. That's why they are colorless. But if you find out that some roots are exposed to sunlight for a very long time, they become greenish. Even yam, when you cultivate this yam, the yam tuber, when a part of it is exposed to sunlight, I discovered that after a while it turns greenish, which means I am of the opinion that plastids might change, meaning chloroplast can become leucoplast, leucoplast can become, I wouldn't know, but that's just my hypothesis. Okay, then we are good to go. Remember that in some organisms, they have this chlorophyll, of course, but it's not in chloroplast, like the cyanobacteria. In the cyanobacteria, their chlorophyll is not in the chloroplast. Remember, cyanobacteria, archaebacteria, is actually a prokaryote, and I'm here describing this organelle here is found only in eukaryotes and never in prokaryotes. But there are prokaryotes that have chlorophyll. Imagine that. That's the cyanobacteria, the blue-green algae. So where are their chlorophyll located? Their chlorophyll is located in the cytoplasm. It's lying freely in the cytoplasm, not in the chloroplast. And remember, the chloroplast is divided into two parts. You have the grana part and the stroma part. The stroma is the part that supports the dark reaction of photosynthesis, while the grana, which is made up of the thylakoid membrane, is the part that supports the light reaction of photosynthesis. You know that photosynthesis is divided into light and dark reactions. So the dark reaction is actually taken care of by the stroma part. That's any other part of it. Yeah, there are stacks of structures called thylakoids, and these thylakoids makes up the grana. So anything outside the grana is the stroma, and that is where the dark reaction and photosynthesis take place, and that of light reaction is taken care of by the thylakoid membrane. That is where you have the electron transport chain taking place, and so on and so forth. Okay, we move straight to other cell organelles. The common one, the ribosome. Ribosomes, what is the function of the ribosome? Ribosomes is actually the site of protein synthesis. So proteins are synthesized in the ribosomes, because that's where, in protein synthesis is the DNA that controls protein synthesis, making use of the three types of RNA: messenger RNA, transfer RNA, and the ribosomal RNA. So ribosome attached to RNA is what assembles, joins the nucleic acid in question, and that leads to the production or the synthesis of protein. So ribosome, simply put, is the site of protein synthesis. But remember, it doesn't synthesize protein alone. It must has an RNA collectively, and that's the joining site where the bonds are formed and amino acids are linked together, leading to protein chain. Then there is another structure called the lysosome. Lysosome. I'm discussing the cell organelles. The lysosomes. So the lysosomes are actually the word lysosome is derived from the Greek word lytic. Lysis means breakdown. So anything lysis in biology is talking about breaking down. So lysosomes are cell organelles that produces lytic enzymes, that is enzymes that destroys engulfed bacteria. And this is playing role in the protection of the cell. So people talk about this when they remember pathogens. Like if you take in disease-causing organisms, what will clear that disease-causing organism from you? If bacteria invades a cell, the cell produces a lytic enzyme. Lytic means breaking down, destruction, that will destroy the engulfed bacteria. And therefore, lysosomes are numerous in most cells that fight against diseases, like the white blood cells, the so-called the phagocytes have enough lysosomes. Understood? Therefore, this lysosome attacks engulfed bacteria. It also attacks the cell. Sometimes it's responsible for the cleaning of the cell, of course. Any worn out cell organelle is dissolved using the lysosome. Then in some cases, there might be misconception whereby the lysosome will start destroying the cells itself. Self-destruction of a cell is called autolysis. So lysosome might destroy the cell itself, and that is what we call self-death, or cell death is what we call autolysis, of course. And then, because of this autolysis, people can cunningly call lysosome suicide bay or stomach of the cell. Suicide bay, it can cause the destruction of the cell. So I thank you for being around. We have many things to discuss, other cell parts, the peroxisome. We have also the centrose, which forms the spindle fiber. The peroxisome is responsible for oxidation reaction, and probably it protects the cell from accumulation of peroxides to cause harm. We have catalase. There are many other cell organelles. I'm happy having you today. It's been a while. It's been nice having someone great like you. I can't imagine I'll get up to such likes on this live stream, but that's great. Tomorrow we'll be here by 3 p.m. also to conclude this free package for you all. Remember, I am here for you in chemistry, biology, mainly. Then biology related courses in your high institution, you consult me. Then mainly I supply laboratory equipments in biology, chemistry, physics laboratory, even your hospital equipments and hospital solutions. You contact me. And then for your quality control in production and advice in production, you contact Her Majesty. I cannot do without you. Then I cannot forget to thank you for helping me get to 10,000 subscribers. Please, let's do more in a majestic way. Then don't stay very, very far away from my community, science community, where my community posts. That's where you suggest topics, because this is where these topics I've been discussing has been suggested. Yesterday live stream was on metals and their compounds. You endeavor to watch them. There are many other educative videos here, your health tips, your DIY, your industrial productions. I cannot mention them, but I cannot do them without you being there. So expand this channel. There are two ways to spread the light. It is either you are the candle producing the light, or you are the mirror reflecting it. Please reflect this channel. Much love from me to you. See you tomorrow. Remain majestic.